What we're going to discuss today is the propagation of an action potential down an axon of a neuron. There are two parts of the nervous system. The first one is the central nervous system. This is the part that consists of the brain, the spinal cord, and the peripheral nervous system. It is named the central nervous system because it sends and receives information from all parts of the body. Secondly, the peripheral nervous system connects the nerves leading to and from the central nervous system by junctions called ganglia. Its main focus is connection to the central nervous system to other limbs and/or glands and creates a communication channel to relay back and forth from the brain to the extremities. The peripheral nervous system is not protected by bone like the central nervous system, which allows exposure to toxins and can create mechanical injuries. First of all, to understand what an action potential going down an axon means, we must understand what a neuron is. The neuron functions as the basic building block for our nervous system. The neuron process is responsible for receiving and transmitting information and also allowing to help communicate with the rest of our body. An axon is an elongated fiber that extends from the cell body to terminal endings to then transmit signals. Most neurons contain one axon, and this is used to transmit information away from the cell body. If we look at this picture, we notice a few things. First of all, we see Schwann cells. These are also called neuromyelocytes, and they're glia of the nervous system, the peripheral nervous system, that is. They are responsible for the creation of the nerve impulses along axons, the nerve development, and regeneration. There are two types of Schwann cells, myelinating and non-myelinating. The myelinating wrap around axons of motor and sensory neurons to make myelin sheath. Also, we notice nodes of Ranvier, which is another term for myelin sheath gaps. They are approximately only 1 micrometer in length and are formed between myelin sheaths that are created by other cells. At this point of the axonal membrane is uninsulated and is able to generate electrical activity. Myelin is an electrically insulating material that creates layers around near the axon of a neuron. Myelin is majorly essential for proper functioning of the nervous system. Saltatory conduction is the propagation of an action potential along myelinated axons from node of Ranvier to the next node. This allows for an increase in the conduction velocity of the action potential. Continuous conduction is a lot slower than saltatory conduction because a nerve impulse travels down the entire unmyelinated neuron rather than jumping between spaces like saltatory conduction. Conduction velocity is majorly an important role in the nerve conduction studies because it is the speed of an electrochemical impulse that is traveling down the neural path. The factors that affect are included age, sex, and also medical conditions. Cells with few branches are called oligodendrocytes. These cells are a type of neuroglia that provide support and insulation to axons that are present in the central nervous system. Neuroglia are also known as the glial cells of the non-neural cells, which help perform functions such as maintain homeostasis, create myelin, and also allow for support and protection for neurons that are found in the brain and the PNS. Another topic we must be able to determine the difference between the voltage-gated sodium ion channels and the voltage-gated potassium ion channels. A sodium channel is an integral membrane protein that forms ion channels. They are determined by a trigger which opens a channel for ions or binding substances. In neurons, myocytes, and types of glia, the sodium channels are what are responsible for the rising phase of action potential. In potassium channels, those are the most distributed type of ion channel and can be practically found in all living organisms. The main function of a voltage-gated potassium ion channel is to open and close in response to the changes in the transmembrane voltage. The membrane resistance occurs when the membrane impedes movement of charges across the membrane. A length constant is the mathematical constant which we can use to quantify distance from a graded electrical potential and how it will travel along an area by passive electrical conduction. Like I stated, the resting membrane potential is -70 millivolts. Let's create a graph that we can describe what I am talking about to see if you can actually comprehend. First off, I labeled the axis with the -70, -60, and the 30. You'll understand what those go for as we go along. It's going to start at -60 as the sodium channels are opening, and it's creating a depolarization as the sodium is entering the cell. Once the sodium channels roughly reach 30 millivolts, they're going to close and the voltage potassium channels are going to open. The 30 millivolts is going to be called our threshold. Once the channels are opening, the potassium is going to go out of the cell, which is going to cause a depolarization. This is going to happen until we reach our axon terminals.